Now, we're going to start looking at lesson 5, but I really don't think that we'll finish it today. We might get through the notes part of it today, we'll see. Lesson 5 is all about multiple alleles. Until this point, we have only considered traits that have 2 alleles. Sometimes one allele is dominant, one is recessive. Sometimes they're co-dominant, sometimes it's incomplete dominance. But there have only ever been 2 choices. That's not real life. That's a few genes that exist in real life, but there are certainly a lot more that will fall into this category we're about to look at. So we're going to start by looking at just the idea of multiple alleles. For example, there is a gene that exists in a clover plant. It has 7 different possible alleles. That means there's lots of different combinations of 2 that you can make, and you end up with 22 different possible phenotypes. That's a lot of stuff. That's beyond what I would expect you to be able to predict, but I just want you to see that there are genes that have lots and lots of different alleles. So that could help explain why, for some traits, there are so many different phenotypes. I don't know what that weird ring says. Okay. But for some traits, there are so many different phenotypes. Now, a thing that you need to remember is this. Each individual, so you or the organism we're studying, only has 2 of these alleles. Is it someone's shoe? Is that what's happening? Oh, it's this phone that I took from someone here. That's what's going on. Okay. Sorry, this is throwing me off. So each individual only gets 2. So even if there are lots of different possibilities, that doesn't mean that you get 7 alleles. You still only get 2. One from mom, one from dad. Even though there are many alleles that exist as possibilities in the population. So we're going to take a look at a gene in humans that has 3 alleles. This is a reasonable thing for you to predict because it's not giving us 22 phenotypes. There are 3 alleles. These 3 alleles are on your data sheet. Now, we haven't looked at it in a while because you haven't had to look up amino acids from an mRNA code in a while, but on your data sheet, which you'll want to locate because we're going to use it a bit more, especially at the end of this unit. It tells you the alleles for blood types. It doesn't tell you how to work out the phenotypes, but it does tell you the symbols for these alleles. So for blood type, that's a capital letter I. And then as a superscript, there's a capital letter A. That's the A allele. Then there's another allele, capital letter I with a superscript capital B. That's the B allele. And then there is lowercase I. That is the recessive allele. It's recessive to both of the other 2. And the other 2, the A and the B, are co-dominant. That means neither can mask the other. They both show up. They're both present. And so with these 3 alleles, okay, oh, someone wishing someone good luck. Can I turn the volume down? I can't seem to do anything. But there are 4 different blood type possibilities with those alleles that we have. A person could have blood type AB. If that's their blood type, the allele combination that they would have is capital I with an A, capital I with a B. So the A allele and the B allele. This part that I added in is not on your data sheet. You have to understand how that works. Now, if you understand how alleles work, you probably don't have to memorize this, but it is something that you would have to be able to work out. If a person had blood type O, which is the most common blood type, they would have 2 copies of that recessive allele, the lowercase I. If a person had blood type A, there are 2 different possibilities. They could be heterozygous and have an A allele and a recessive allele. Or they could be homozygous and have 2 copies of the A allele. Very similar story for type B blood. A person could be heterozygous and have one B and one recessive allele, or have two B alleles. Now you'll notice I didn't include the positive or negative here because that's a whole different gene. And this is only one way of typing blood. If you remember from Bio30, it all depends on when you took it, blood types are based on antigens that are found on the surface of red blood cells. And there's actually another way that you could type people's blood. This is just the ABO system. So just keep that in mind. This is just one way of typing blood. Now, side note, do any of you actually know your blood type? It's something that people don't generally know unless they've needed to for something. If I had to guess, you probably have type O because it's the most common blood type. But because it's determined by these three alleles, if you knew what your parents' blood types are, you could probably predict what your own blood type is. Now we're going to do a few examples to show you how this would look if we're doing crosses. Keep in mind this second point though. Even though there are three alleles, each individual doesn't get all three. Each individual only gets two. So we're going to take a look in my first example at the phenotypic ratios predicted in the offspring of two people whose blood types are A, B, and O. And so the first step in any cross question is to figure out what the parent's genotype and phenotype is. The question is telling me the parental phenotypes, so I need to figure out what genotype that is. Put those genotypes as gametes by using the law of segregation, and then predict what goes in the 4 squares or potentially less of my Punnett square. So before I go to the screen that has my answer that will pop up, I want to give you a minute to think about this yourself and see if you can set up a Punnett square for this. So I'll pause here. Now if you'd like to look, I put my answer up on the screen. So I started over here, kind of on the left, by writing down what the parents' genotypes are. So the parent who had AB blood, that's their genotype. The parent who had type O blood, that's their genotype. For the parent who had type AB blood, they had 2 possibilities for gametes. For the parent who had type O blood, their 2 possibilities are the same. So I didn't bother making a 4 square Punnett square. I only made a 2 square Punnett square. You'll see that the ratios or the percentages you get would be the same if you did 4 squares or 2. What you should be finding is that 50% of the time, offspring should have type A blood. And 50% of the time, offspring should have type B blood. Now you might think this is weird because neither parent had type A or type B. One had O, one had AB. But because there are multiple alleles, there are more than 2 phenotypes, and it's very normal for kids to have different phenotypes than their parents, especially when there are multiple alleles controlling that particular trait. So in this scenario, none of their kids would have the same blood type as them, even though that they are the biological parents of these kids, just because of segregation, Mendel's law there, and that each parent is going to provide one allele to their kids. None of the kids will have the same blood types as their parents.
Sorry, this might be a dumb question. Can you explain segregation?
Sure. The law of segregation is Mendel's first law, and it says that when gametes are forming, you get one and only one copy of each gene. And we could relate that further back to the last unit, because we know that you get one copy of each chromosome, and genes are just sections of that. So segregation says that when we're forming gametes, these are the 2 possibilities. Either the gamete will have the A allele or the B allele, but it wouldn't have both because those are both coming from the same parent. Each parent has to be providing one copy of each gene to their offspring. So we can't have 2 copies of the gene coming from the same parent. That would go against the rules of segregation. Does anyone else want to ask anything there? Okay. So we're going to do another example. And this time, I'm adding something to the question. I I've got a man with blood type B and a woman with blood type A, and I'm telling you that they already have a child whose blood type is O. Now, why would I tell you that? It's to give you a bit of information so you could actually figure out their genotypes. But then when I ask you the question, I want to know not only what are their chances of having a blood type A child, but also that it's a girl. Now, yesterday, we took a look at questions that were sex-linked, that were on the X or on the Y chromosome. Blood type is not attached to the X or Y chromosome, so we have to consider both things. And I want to show you something that you can do that might make your life easier. So I'm going to start at the beginning with mom and dad's genotypes. It said there was a man with blood type B. So here's my man. I added the X and Y chromosome, not because of blood type, but because the question is asking me to predict the chances of having a girl. So I know that I need to understand how sex chromosomes are being passed on. He has blood type B, and at first, I had a little line there. I wasn't super sure what his second allele was. Having blood type B could mean that he was homozygous for the B allele or that he was heterozygous. There was a woman with blood type A, so I put the two X chromosomes because the question was asking me about the sex of the baby later on. And then for blood type A, I would have done the same thing. She has to have one copy of the A allele, but I wasn't super sure what her second allele would be. Since the man and the woman had a child already who had blood type O, blood type O is those two recessive alleles. For that child to have gotten two copies of the recessive allele, both parents must have had it. We've used this logic before if you think back to like the first and second lessons. There was questions about cats and about horses, and sometimes I would tell you about their parents. And the reason I was telling you about their parents is so that you knew that they were heterozygous for a trait. Now, up until that point, does anyone want to ask a question there? Okay, notice how the recessive trait is always the thing that's giving me answers. Since they had a kid already with the recessive trait, they both had to have the allele. Even if they didn't show it, they had to have it. Now, since in this question I'm considering two things, blood type and sex of the baby, I don't actually have to make a big, massive Punnett square. You could, but I find those clunky. When you have traits that are on different chromosomes, and it'll tell you in the question if something is sex-linked, so that's not something you have to memorize or guess. But blood type is not a sex-linked trait. It's not attached to the X or Y chromosomes. So when you have two traits that are on different chromosomes, you can do two tiny Punnett squares and combine them. That's actually my preferred way for doing a Punnett square or Punnett squares when I have more than one trait that I'm considering. So if you take a look at what I did here, first I made a Punnett square for blood type. I took just the alleles for blood type. So the man had a B and a recessive, the woman had an A and a recessive. So I put their possibilities on each side of the Punnett square, and I figured out that they had a 1 in 4 chance of having a child with type A blood. Now if I look, the first one isn't type A, that's type AB, which is different. On the bottom, I would get type B or type O. The other part of the question is what's the sex of the baby? Now, you could make a Punnett square for this, or you could just remember it's always 1 out of 2. The chance of having a boy or the chance of having a girl, always 1 out of 2. See how I made the Punnett square here? I didn't even have to include the second X if I didn't want to. I just did so that you could see where it was coming from. But without making a Punnett square, I think it's fair and reasonable for you to say there's 2 genders, the chances are 1 out of 2, always. And you wouldn't need to make a Punnett square if you were going to say that in a question. You could just say, I know the chances are 1 out of 2. Then I take those two numbers and I multiply them, not add, multiply. So I got 1 out of 4 for blood type, 1 out of 2 for girl. So I went 1 out of 2 times 1 out of 4. When you multiply fractions, you just multiply straight across. Denominator times denominator on the bottom, numerator times numerator on the top. That gives me 1 out of 8. The percentage for that is 12.5, or if you round it up, it's 13%. And if you were going to put a probability between 0 and 1, it would give 0.13. Now to me, that is simpler than having a Punnett square that has like 16 boxes in it. I wouldn't prefer that. If you like that, you can certainly do that. But when the genes are on 2 separate chromosomes and independent assortment is happening, which it will be the vast majority of the time, this I think is a simpler strategy. Does anyone want to ask anything there? We got a couple more scenarios. I think maybe I'll talk about this one and we'll save the other one for the next time. But in this scenario, sometimes when there are multiple alleles, instead of what we had with blood type where it was just dominant or recessive, we get what's called an order of dominance, where one allele is the most dominant, then another one is like second, another one is third, another one is last. So there's a gene that controls rabbit fur color that has 4 alleles. You don't have to memorize these details. This is the kind of information that would be given to you in a question, but you would have to understand how it works. Agouti is a particular fur color pattern. It's controlled by the most dominant allele, so it got a capital letter. Then I used the greater than sign to show you that it was more dominant than the chinchilla fur color pattern, which is more dominant than Himalayan, which is more dominant than albino. Oops, that should have a little C next to it. Albino, but I tried to write it on the smartboard, remember, so it wrote it 15 centimeters away from where I actually wrote on the smartboard, because it's a dumb board. Oh, yeah, I know we have a Thanksgiving liturgy, thank you. So if I take that information for those alleles, I could figure out which genotypes would give me each fur color pattern. So for example, agouti would be any genotype combination that had a capital C. It wouldn't matter what the second allele was, because the agouti allele is the most dominant out of the 4. For the chinchilla allele, it couldn't be paired up with agouti. If it was, the fur color pattern would be agouti. But if I had 2 chinchilla alleles, chinchilla and Himalayan, or chinchilla and albino, any of those combinations would give me a chinchilla fur pattern. That's because chinchilla is more dominant than Himalayan and albino. I could do the same thing for Himalayan. It couldn't be paired up with agouti or chinchilla because it's less dominant than them, but it's more dominant than albino. So that means that if I had 2 Himalayan alleles or 1 plus an albino allele, I would get a Himalayan pattern. And the only way to get the albino fur pattern would be to have 2 copies of that most recessive allele. So if you consider this, the thing that we're changing is the alleles. We're not changing the way a Punnett square works. That part actually has been the same since the beginning. We're always using the law of segregation. Unless the question very specifically says you can't, you're always using the law of independent assortment, where one gene doesn't affect another, and they're separate from each other. So the only thing that's changing is the alleles that we have and how we interpret the phenotype. So I think what I'd like to do, and I'll give you a few minutes to think about this, then I'll show you the answer, and this is probably where we'll end. We're going to take a look at this question. I have an agouti rabbit and a chinchilla rabbit, both of whom had an albino parent. So think back to the last question. Why would I be telling you about an albino parent? It's probably because there was some uncertainty with their genotype, and this is going to help clear that up. I'm going to cross those 2 rabbits, and I'd like to know first, what are the chances that the offspring has chinchilla fur color? And then, what are the chances about having chinchilla fur color and being male? I didn't say it here, but sex determination works in rabbits much like it does in humans. And questions will normally state that, especially diploma questions, just so that you're confident that it works the same way. So I'd like you to take a few minutes. All right, I'm gonna have to say something about this because I have to shut it down pretty soon. If you take a look, we end up with kind of the same scenario we did for blood type, but this is a different allele. If I consider the genotypes of the two parents, my agouti parent had to have the capital C, but because one of their parents was albino, I know their second allele had to be the recessive one. If a parent is albino, that's the only allele that they have, and because of the law of segregation, each parent has to pass on one and only one copy of the gene. So I know that the second allele is going to be the little c. For my chinchilla rabbit, it's sort of the same scenario. They have the chinchilla allele, but their second allele is the albino one. So I set up my Punnett square like this. I find that one of the 4 boxes gives me chinchilla fur color. The first box does not give me chinchilla, even though I have that allele, because I also have an agouti allele there, and it is more dominant. So even though the chinchilla allele shows up, that's not the phenotype the organism's going to have. So I got 1 out of 4 chances, or 25% or 0.25, of having chinchilla. And then if I want chinchilla and male, I'll take that 1 out of 4 and multiply it by 1 out of 2. I could make a Punnett square, but I know that for male and female, it's always 1 out of 2. There's only 2 choices. So I go 1 out of 4 times 1 out of 2, and it gives me 1 out of 8, just like the last example did, which is 13%. It's really 12.5%, but rounded it's 13. And as a probability between 0 and 1, it's 0.13. Now we're not going to finish this lesson today, so I'm not going to ask you to do the practice problems for homework today. You certainly could try, because you can do lots of them. In fact, you could probably do most of them without me finishing the lesson. So if you want to get started on them for homework, super duper, but they won't be official homework until later.